My name is Zachary Hunter, and I study multi-omics of Waldenstrom's macroglobulinemia here at the Dana-Farber Cancer Institute. And the reason why I'm drawn to multi-omics is because it really allows you to get a full picture of the biology of the disease. And this is how we're able to really advance therapy and improve therapy. Studying multi-omics gives us a great deal of insight into the biology of the disease. And this is important because it's really the biology that drives therapeutic development. If you think about it, we can't develop or fix problems that we don't understand. And why multi-omics in particular is helpful for this is imagine looking at a picture where you can only look at individual primary colors at a time. Normally, we see the full spectrum of color, but imagine you could only see red, right? You get a lot of information out of that picture. And when you start adding in these other colors, you know what? Some of it's going to be exactly as you expected, but some things are really going to change and really start to pop out. And this is sort of how multi-omics works. As you start layering these different types of data, things become increasingly clear. And certain things that once looked sort of insignificant and not that interesting, all of a sudden pop up as these bright colors and really interesting points of data that we can then use to develop new therapies to target this rare disease. So what are the kind of things that we work on? So we work a lot on DNA, and you can think of DNA as the instructions that allow the cells to make tools for themselves, right? And when the DNA changes, it's changing how those tools are made. And sometimes that makes a tool work better. Sometimes it makes it work worse. Sometimes it changes the way it's regulated. So when it's used or not used. And this is really interesting, but it's only one piece of the picture. For instance, we can have a very interesting looking change in the DNA in a gene that turns out is never used in Waldenstrom's, in which case it doesn't really matter, right? So it looks really interesting from the DNA perspective, but from the perspective of Waldenstrom's overall, again, with this idea of we're keeping adding these layers or colors in, it may in fact not be that interesting. We also look at RNA, which is how these genes are used in the form that they take. So if we stick with this idea of genes being tools, let's imagine a hammer. So we have a gene that encodes for a hammer, but genes in sort of higher mammals are actually pretty complicated, and they don't just have one form, in the same way that hammers don't just have one form. There's small hammers, there's big hammers, there's sledgehammers, right? And hammers have different parts to them, right? You know, there's the part obviously for hammering nails, but if you look at the back end, there's also a little fork that allows you to pull nails. And this is actually a really common thing in genes. There'll be a form of a gene that does an important function like hammering nails, and a part of the gene that actually does quite the opposite, takes the nails back out again. And this allows some local regulation of a process, just even within the gene. So we can look at how the gene is being expressed and used. And we can also look at what form of usage that we're seeing. Are we making sledgehammers? Are we making mostly things that are pulling nails? Are we making regular hammers? How much of it are we making? How does that line up to these changes we saw in the DNA? And then finally, we have how all this is regulated. How does the cell know when and what and how to use, in what context, all of these different tools? And this is called epigenomics. And if you think about it for a minute, the DNA in your body is the same throughout, in your heart, in your brain, in your blood, in your toes, everywhere, right? But all these tissues are really different from each other, really, really different. And they respond differently to the exact same set of stimuli. And this is because of epigenomics, the regulation of the DNA itself. It's what parts of the DNA are cells allowed to see. And by altering this, we can effectively make sure that each tissue actually sees only a limited part of the genome and effectively has its own private tool set that's different from all the other tissues. That allows for a great deal of diversity and both of response and in terms of what these tissues are allowed to do, which is why they all act so different from each other, even though this technically the instruction set is all the same. And in cancer, not surprisingly, this goes completely awry and cells gain access to tools that they're not supposed to have and lose access to tools that they desperately need. Again, we can sort of think about this as, as these multiple different ways of fine-tuning a response. So let's say, sort of take macroscopic, let's say your DNA, we have instruction sets on how to build a house, right? And how to just build things in general. But when you're going to build a house, right, there's a certain set of tools you need. And what will happen is a signal will come in and be like, we're building houses now. Let's turn on all the tools that are going to make houses. And what we can then do is we'll see all these different genes get turned on again. But it may be at any individual moment, right? Maybe, for instance, at this stage, you don't actually need hammers. In general, for building houses, you need hammers, but right now you don't. And so what you see at the epigenomic level is all the house building tools get turned on. And at the local level, individual genes will be switched around, either between different forms, again, different kinds of hammers, or even maybe making no hammer, right? The gene, they'll look at the gene and they'll read the gene, but the actual new instruction for a particular form of that gene is actually to do nothing, because you want the ability to turn on all the tools that you need for houses, but maybe at this particular moment, not this particular tool, right? And this way you can fine-tune things globally and locally in a very, very fine way. And so if we hope to understand cancer, we need to be able to hold all of this together, right, at the cellular level, and find out what it is we can do to identify the key points that the cells really, really care about. And I think the thing to remember here is that the cells are fundamentally sick. They're not supposed to be doing what they're doing. And on some level, they know this, right? And so there's some part of them which realizes that something's gone wrong and really in some ways wants to die and wants to, so it doesn't cause problems for you, right? the larger organism. How can we identify these things? How can we amplify that signal and suppress the signals that the cells have adapted to overcome this in cancer? And ideally, if we can get it right, we can hit these cells, the cancer cells, without hitting the rest of your body. And, because therapy, sort of famously, it's like trying to repair a car while the car is still running. We have to be able to go in and take out these cancer cells without hurting or permanently hurting the person. And this is why we use multi-omics, is it allows us to better understand what the biology is and how we can interfere with that biology in a way that is specific to the tumor and what's going on, what's causing the problem, and minimizes the impact in the rest of the body. The other thing that it allows us to do is try to understand the processes associated with the disease. Now, this can be something like disease progression, the location of the disease. Is it in the spleen, in the bone marrow, in the lymph nodes? How fast is it growing? Whether it responds to a particular therapy. Some therapies seem to work no matter what. Other therapies really are specific to different versions of the cancer. And one of the other things we can do with multi-omics is sort of start coming up with natural groups or clustering of these of cancers. You can think flavors of Waldenstrom's in this case. And in our case, what we've discovered is that there are basically two major flavors of Waldenstrom's that sort of evolved from a common precursor. And each one of these sort of has its own set of characteristics, the way that it presents to the patient and to a clinician. and respond slightly differently to different therapies. And we can also use multi-omics to track patients' progression along this stage from sort of early diagnosis to the disease actually sort of rapidly expanding in the marrow and needing therapy. And because we can model this, we can also try to understand what are the biological processes that allow this to happen, right? So not only can we say prognostically, okay, we think that you're going to be okay for a while, or we think that you're going to need therapy soon, but we can also go back and we can say, okay, how did this happen? And is there a way to block it, right? Can we block this expansion in the marrow? Can we roll it back to an earlier version of Waldenstrom's that might be more stable? And this is both the goal and what we're actually accomplishing in many ways in our current studies in multi-omics of Waldenstrom's.